I just want to introduce myself and say what a huge pleasure it is to be able to come and talk to you all. I don't know you as a group, and I find that quite odd. Normally I'm coming to give these talks to groups that I know well. I run a clinic at Great Ormond Street for children with retinal dystrophies, and just by a quirk of history, the only group that is not represented in that clinic is the children and families with Bardet-Biedl syndrome, who happen to be have been historically looked after by my colleague Will Moore, so some of you will know him. So thank you for welcoming me, and my plan today is to assume, maybe blithely, that there is relatively little knowledge about ophthalmology. The reason I assume that is that most of my medical colleagues know nothing about ophthalmology, and so I'm going to start with the basics and just give you a little guided tour to what we do in clinic, what those things that we talk about in our complicated letters actually mean. And then I'm going to go and talk a little bit about gene therapy and about some of the other technologies, research that we're using to try to either prevent blindness or maybe even restore vision at some point in the future. It's a very general talk. It's not going to hone in very specifically to Bardet-Biedl, but I imagine that you're going to have a ton of questions, and I'm more than happy to talk about those in the Q&A later. So let's start at the very beginning, the eye, and I'm just going to quickly rattle through some of the anatomy, some of the terms that you almost certainly know, and so forgive me if you do. So we start at the front of the eye with the cornea, the clear dome on the front of the eye. Okay? Behind that is an area called the anterior chamber, which is delineated by the iris, the coloured bit of your eye. The black dot in the middle is your pupil. And sitting immediately behind the iris is the lens. The lens does the fine focusing of light. That light travels through the vitreous cavity filled with the vitreous humour, a jelly-like structure, and the light hits the retina, the photosensitive layer of the eye, which has a number of different cell types in it, but the one that, as Phil was alluding to earlier, the ones that you are most interested in are the rods and the cones for your daytime vision, the cones for your detail vision, the cones for your colour vision, and your rod cells, the cells that allow you to see in low illuminations. And the rods and cones, as you may be able to see, and I'm going to describe what's on some of my slides because I'm aware that many of you can't see what I've got up here, but the rods and the cones have a couple of different components to them which are important for when we're thinking about therapies. So they have the cell body, or what we call the inner layer, and then they have the outer part of it, which is the outer segment, which is where the process of turning light into an electrical signal happens. And for those of you that can see, you'll see at the top of this cell here, this outer segment, it's a series of pizza slices, if you like. And those pizza slices get refreshed every night, and it's there that the light is turned into an electrical signal. But it's also the bit that is first lost when you have a retinal dystrophy. And it's important to remember that because as Phil alluded to earlier, what we're starting to see in some of the trials that I'm running is that whilst the outer segment, the pizza slices, are lost, the inner segment, the cell body, sometimes remains for quite a considerable period of time, and there is some evidence that you can get restoration of the outer segment with gene therapy. So what do we do in clinic? We have a number of different investigations and a bewildering array of different devices that we try and shove you onto as technology allows us to probe more and more functions. And we're all quite geeky as ophthalmologists. We like our toys, as you may have seen. So we have lots of things that we try and use to understand disease better. The first thing that I think is important to understand is what on earth are we measuring when we're doing vision, when we're testing your vision. Now, vision, visual acuity, as we call it, is not what you're wearing in your glasses. It's the most common misconception is when people tell me, oh, I've got terrible vision, doctor, terrible vision, I've got these enormous thick glasses. I don't really mind about your glasses. We can always correct your vision with glasses. It's a bit like wearing a pair of shoes. I don't care what size shoes you wear. What I care about is how much you can see when you've got your glasses on. And so you'll see in the letters that we write to you, we mention visual acuity, and there are very confusingly a number of different scales that we use. So the one that everyone really is probably most familiar with is the so-called Snellen scale, which is a fraction. And there are two ways of expressing that fraction. So you might have heard of, he's got 20/20 vision. And what that means is that standing at 20 feet, you can see what a person with so-called normal vision can see standing 20 feet away. What happens if you had 2200? That would mean that if you stand at 20 feet, you can see what someone with normal vision can see standing 200 feet away. Now, we don't really use feet in this country anymore, thank heavens. We use meters. So the same is 6/6. 6/6 is the same as 20/20. 660 is the same as 2200. In Europe, they use decimal. I have no idea what decimal means, so we'll forget about that. So most commonly nowadays, we use a chart called logmar. And logmar 2020 is 0. Logmar 2200 or 660 is 1.0. So you get a number anywhere between that. The closer to 1 you are, the worse your vision, and you can go over 1, you can go up to 2, in fact. So most of the letters that you'll see from us express visual acuity as a logmar value. Okay, moving on. Other tests, visual field, and we have lots of different ways of measuring visual field. And for those of you that are likely to be enrolled in the trial, we will try and get visual field measurements, and visual field is really important to you, as you all know. Being able to see, to avoid tripping up over things is hugely important. And then we have other ways of looking at the structure of the eye. So what I'm showing on the slide at the moment is a little slit lamp, which we use for examining smaller children. And then when you're bigger, we can stick you on the bigger table-mounted slit lamp. Sometimes I like to use a little hat on my head. And what you're seeing here is me using an indirect ophthalmoscope to examine something that's lying on a table. In fact, that something that's lying on a table turned out to be rather larger object lying on a table, which is a male silverback gorilla. So I'll stop and just divert. So part of my training, I did a year of training in Canada and a year of training in Australia. And one day it was in theatre in Australia when I got a call going, "Oh, g'day, is that Dr. Henderson?" And I went, "Yes." "Hi, it's Rachel from Werribee Zoo." "Oh, yes. Now, look, you're the local primate expert, and we wonder whether you'd come and have a look at a gorilla of ours." Which I was entirely delighted to do, and I insisted that the only way I'd do that is payment would be allowed to bring my young children and for them to come and have a look too. And so I duly cancelled the clinic gleefully and went to see this gorilla. And so what I realized is that the person that accompanied the gorilla, which I've got a photograph on the screen at the moment of a large chap sitting there by the gorilla with a very large gun, which was a little disconcerting, and I subsequently think in latter years that it would be quite useful to have that same man sitting in some of my clinics. So we mentioned imaging, and imaging has increasingly become a very important part of what we do, and it allows us to see in incredible detail how disease is progressing. So we have a camera now, an Optos camera, which allows us to see in a single shot 200 degrees of the retina, and we can image both in color and what we call autofluorescence, which is a black and white image, what is working and what is not working. And I'm showing a picture on the screen of a patient who's got large areas of retina that are not working, and you can see that in that autofluorescence. We often inject dyes into you. I can turn you the colour of a dodgy Donald Trump, as I say to some of my patients. And then we have this tool called optical coherence tomography, OCT, and that allows me to see at almost cellular level what's going on in the retina, and I can see that in cross-section, and that's a very important part of this trial because that's going to allow us to see whether the photoreceptors are still present and whether our gene therapy is potentially working, whether it may even be rescuing a little bit. And we can image vessels, and we can do all sorts of things in extraordinary detail now, which is an amazing place to be. You'll remember having electrodiagnostics as well. My colleague, Dr. Thompson, many of you who've been through Great Ormond Street may remember, where we stick lots of little sticky electrodes all over your head and on your cheeks, and we can probe the function of those photoreceptors. We can see whether they're working or not, and by flashing different lights and making you watch TV, and so that's very useful. And then at the end of it, we've got a diagnosis potentially, but we need to establish what gene it is. And so you, of course, probably all know your gene thanks to the work of Phil and Elizabeth. But for most of the patients who come through my clinics, we're starting at ground level. We've got to give them that diagnosis that they have a retinal dystrophy, and then we've got to try and understand which gene it is that causes that retinal dystrophy. And there are now over 270 different genes that can cause retinal dystrophies. So it's an ever-growing number, and there are all sorts of terms, not just Bardet-Biedl, but there's senior Loken syndrome, and there's Usher syndrome, and there's Joubert syndrome, and there's Leber's congenital amaurosis, but they're all genetic disorders that cause loss of vision secondary to a retinal dystrophy. So where are we now? We have genetic testing, and genetic testing has rolled out, and actually we're in an incredibly fortunate position. We're able to test in the UK far more easily than nearly any other country in the world, actually. It's one of the things that we do really well in this country. And so through whole genome testing, we've moved to being able to give you and patients like you a genetic diagnosis probably about 70% of the time. Now, when I first started, I'll take you back on a bit of a history tip, we used to what we call sequence one gene at a time. So what I'm showing on my slide at the moment is something called an electropherogram. This is work that I did during my research. This is back in 2004 when I first started, quite a long time ago, research in retinal dystrophies. And I was involved in the first gene therapy trial, which was for a condition, for a gene called RPE65. And I would take patients who had an early onset retinal dystrophy, and I would sit there laboriously trying to take their DNA and look for changes in RPE65. And we found a cohort, and we got a cohort together. And so fortunately, that technology has been superseded. Although we still used gene by gene sequencing, we now are able to, as you know, do massive parallel sequencing of lots and lots of genes all in one go. So we now are able to provide you with a diagnosis, and we're getting to the stage now with all of these different gene therapy trials where more and more patients have the option of potentially of being enrolled into a trial. Now, Phil's already talked about what is gene therapy, so this slide is largely irrelevant. But just to recap, the goal is to get a healthy copy of the gene into the cells that are still alive and still hanging in there, and in order to try and stop them from degenerating. And we do that by putting that DNA into a virus, as you've heard. And so what I'm going to show you now is, and forgive me for those of you that can't see, but I'm going to show you some video of what subretinal gene therapy looks like. So it's a fairly straightforward procedure compared to a lot of the things that I end up doing. And what I'm showing on the screen at the moment, hopefully it's going to start playing, I go into the eye through 3 tiny little incisions that I make in the wall of the eye, in the white part of the eye. I remove the jelly from the eye, and that's called a vitrectomy. And then I inject, using a very tiny cannula, under the retina. I make a little injection through the retina, and I make a little blister. And I'm lifting the retina off and injecting fluid containing a gazillion copies of these viruses. And I might do 1 bleb, as it's called, or I might do several blebs. And blebs, they're funny. They don't behave in an entirely predictable manner. And the more what we call dystrophic your retina is, the more advanced your retinal dystrophy, the less predictable the bleb's behavior is. So some of the videos that I'm showing show blebs that are behaving really beautifully, and they go under the center of the vision in one nice smooth injection. And other times the bleb behaves entirely erroneously, and it goes completely in the wrong place. And I'm showing one video where the bleb decides to go up and around the optic nerve in a very strange way. But in the end, my goal is to try and get the center of your vision, the macula as we call it, treated so that the photoreceptors are exposed to the virus under the retina. And then that bleb will slowly settle down after 24 to 48 hours. And I fill the eye with an air bubble, and that air bubble helps the wounds that I've made seal. And the air bubble slowly dissolves away after about 5 days, and the retina settles back down into place. And hopefully all those cells have now got a copy of the gene of interest. Now, what I'm going to show you now is 2 videos. So at the moment, there is only 1 licensed gene therapy. And that was for that first gene that we treated back in, we started those trials in 2006. That trial data was published in 2008, I think it was now. It took until 2017 for the FDA to license it, and I started treating NHS patients with it in 2019, just at the beginning of COVID. This is one of the earliest patients that I treated. His name is Haran. His parents are very happy for me to show his videos because they demonstrate the power of gene therapy. So the first video, I don't think we're going to have any sound, sadly, or if we can, you can turn up the sound, that would be great, is of a little boy. He's 2 years old, and his father is asking him, where is the door to the room?
Where's the door?
And he's slowly feeling his way down the side of his cot, trying to find the door.
Where's the room door?
And this is a room that is normally lit with an overhead light. And children with RP65 can barely see anything except in bright sunlight or in bright light.
Where's the door to the room?
And he's feeling his way now along the wall.
It's the wall, yeah.
And eventually he feels the door. But for those of it that you can see, I think all of you would recognize that this is a child with very limited vision. The next video I'm going to show you is what he's like after gene therapy. He's now kneeling on the floor about a year later.
And Mummy's gonna come today.
And he's picking up polystyrene beads from his beanbag off the floor with perfect dexterity. It has been utterly transformative. And it always makes me slightly emotional seeing this film because he's done amazingly well. Not everyone has done as well as that. And to tell you that it always goes entirely smoothly would not be truthful. As Phil alluded, gene therapy has its complications, and the ones in the eye that we're most worried about are of inflammation, the body's immune system reacting to the virus, and that inflammation causing damage to the retina. And actually, we didn't discover one of the main complications in the trials until after it had been licensed, a complication called outer retinal atrophy, where patches of retina just die away probably because of that inflammation. And so we have had 1 or 2 children who have actually lost a bit of vision because of inflammation. So we have changed our steroids regimens, and we hope that we're getting better at controlling that, but it's certainly something to consider. So I'm going to rattle through now, because I'm aware that I'm probably running over in terms of time, some of the other therapeutic approaches. Some of the challenges with gene therapy are that the genes that we can only use at the moment are for small genes, because the current set of viruses that we use can only really take small genes. One of the approaches is to cut bigger genes into two. So I'm about to start a trial for a condition called Usher syndrome, where we cut the gene into two pieces, fill those two pieces into two different viruses, and stick both copies under the retina, and then hope a proportion of the cells will join the two bits up together and make a full-length protein. So we'll see if that works, and that's one of the new approaches. One of the really exciting, very early stage trials that we're seeing is for a type of therapy called optogenetics. So when you've lost all of your photoreceptors, you no longer have the ability to turn light into an electrical signal. But we know that there are proteins used by a number of fairly primitive organisms, often found in the depths of the ocean, where they have proteins that can turn light into an electrical signal. And so what we've started to be able to do is to take some of these proteins, which are called channel rhodopsins, put them into viruses, and infect other layers of the retina, so that suddenly you might be able to turn other cell types in the retina into kind of basic photoreceptors. And there are now a couple of trials of optogenetics, and there are quite a few hurdles still to overcome, and we probably haven't got quite the right channel rhodopsins that yet to really make it effective. But some of the early data is very exciting. And we've certainly seen patients who've had really no light perception now suddenly being able to, with devices, so they have to wear a pair of goggles in order to turn, to illuminate these channel rhodopsins properly. But being able to take people who have got no light perception and actually having some very basic form vision. So certainly it's an area that I think we'll hear more about in the future. Now, one of the other areas that I'm involved in, and this is a little bit further out, but offers hope again for people who've lost all their vision, is stem cell therapy. Now, I'm not talking about the sort of stem cells that you'll see advertised in parts of the world where science is a little bit more dodgy. And the United States, for example. Actually, I say that slightly in jest, but I actually mean that. You can do some extraordinary things in the US, and as long as you're not too afraid of lawsuits. So in Florida, there was a group that they would make you pay for the privilege of being part of this trial, and some of you might have seen a BBC documentary based on this, actually. But they were taking stem cells from your bone marrow, spinning them down and then sticking them in the eye, and charging you 20 grand for the privilege. Surprisingly, they got some pretty awful results and wrecked one or two eyes in the process. So I say that as a caveat to saying, for all of you that understandably will be looking for therapies all over the world, do treat a lot of what's out there with a healthy degree of skepticism. The sort of stem cells that we're talking about, though, so my colleague Jane Sowden, who works at the Institute of Child Health, and I are about to start large animal model trials where we, first of all, take a skin a biopsy. We can take some of the cells from that skin biopsy, and we can reverse engineer them and turn them back into a basic stem cell. A stem cell essentially is a cell that can be, given the right cues, turned into any cell in your body. Now, what we want to do is turn that reverse-engineered stem cell into a new little eye. So we can take your cells from your skin biopsy and turn it into what we call an eye cup. And in the lab, we can grow up little tiny eyes in the cell based on your DNA. Now, rather than take yours, we might take one from an embryonic stem cell from a healthy person and grow up healthy retinal tissue. We're now able to print at a micron scale wafers that we are then going to take photoreceptors from those eye cups. We're going to seed that wafer with rods and cones at a density approximating the human retina. And later this year, we will be implanting these wafers under the retina in a large animal model to see how the retina responds to having these rods and cones under the retina, and see whether or not they start to integrate into the retina and maybe even start functioning. So there is, obviously our hope is that that will eventually someday be a treatment for those people who have lost all their vision. We're a long way away from that, but it is at least a start, so a cause for optimism. So I think that's probably enough from me. I'll wrap up and hand over to Elizabeth. Hopefully that's given you some idea of what we do and where we're heading, and I'll welcome questions in a little bit. Thanks very much.